Hi everyone, welcome to my new webinar. Today our guest is Professor Donald Pfister, who is a mycologist from Harvard University, and he is well known for his extensive research on fungi and also for his decades of teaching and leadership at Harvard. He has served as the curator of Harvard Herbarium. Thank you, Professor Pfister, for joining this interview. Your insights are very valuable to us.
Thank you. Happy to be here.
Okay, so I'm going to start with the first question. So I would love to hear your own story and how you got interested in fungi. Was there a specific experience, mentor, or like a discovery that inspired you early on?
Well, I began my career, I suppose, in as an undergraduate at Miami University in Oxford, Ohio, and that there was a botany department there, and I decided because I liked plants that I would be a botany major. And within the department, there were only a few professors. It was a very small department. And the professor who was teaching the mycology and plant pathology course was William E. Wilson. And Prof. Wilson was very intimidating. He made sure you did all your work the way you were supposed to do it. But he was very interested in fungi and exploring about fungi and so forth. He wasn't a researcher, he was a teacher. And my first real experience with a fungus and with Prof. Wilson was when I went out near the dorm where I was living in the woods and I picked up a bright red fungus, this genus Sarcocypha, probably what we would call Sarcocypha dudleyi today. And I was very naive. I thought, oh, nobody's ever seen this before. This is just fantastic. Wow, and so I took it in my hand to Prof. Wilson, and his comment was, oh, you found that. And he got out a handbook and he showed me what it was. And I just thought that was fantastic that I could go out and find something and we could identify it, even if it wasn't new. And so I just kind of got hooked and went to graduate school and continued my studies there at Cornell.
Mhm.
So a mentor.
I think my interest started in childhood. And similarly, my mom actually took me on a foraging walk when I was a kid. And I remember just like the idea of looking behind plants and trying to find mushrooms in hidden places just seemed very intriguing. And
Uh-huh.
I feel like as I grew older, the same curiosity carried on.
Yeah. Well, it's curiosity. Curiosity is really important in doing these things, and fungi lead you to be curious in many ways.
So I read that you're specifically focusing on Escomycetes, and I was wondering why specifically you became interested in this group. Like, what makes them so special compared to other fungi?
Well, I don't know that they're special compared to others. I think I was intrigued by them because some of them, a lot of those that I study, the Pisizales are beautiful
Mhm.
and colorful. And when I began studying these, we really didn't know a whole lot about their biology. So it was always, you'd mentioned curious. I was always curious, what are they doing? How are they getting along? And I was also happy and intrigued by being able to study the microscopy rather than just looking at the fungus and giving it a name. I was always happy to section it and look at it, try to identify it with its morphology. It was also often possible to culture them so you could grow them out on a petri plate.
Mhm.
And that gave kind of another set of hints about what it is, what the biology of those fungi were. So a combination of things, beauty and fascination with the microscope and then being able to grow and cultivate them and think about them that way.
For sure. I read about a lot of fungi species, but not much about Escomycetes. So that's definitely on my list.
Yeah. Well, I don't know if you came across my papers from a number of years ago on the nematode trapping fungi. You may not have seen that.
No.
Um, it's been known that for a century or more that some fungi can trap and consume nematodes, little worm structures in the organisms in the soil. And that phase of the trapping is the asexual phase. And nobody knew how they reproduced sexually. And purely by accident, I was working on culturing some of the fungi that I had access to. And I cultured one of the orbilias. Orbilias are little tiny discomycetes. They're very difficult to study sometimes. But I cultured one of them and realized that what I had cultured and come up with in culture, and I recognized it as being one of these fungi that trap nematodes. And you can only see the traps and see the trapping mechanism when you add nematodes to the culture.
Oh.
So I had grown up the fungus, I got a supply of nematodes, I added the nematodes, and they produced the ring-like traps that these produce. And so that led to a whole series of papers and work on these fungi. But it kind of demonstrates here's the microscopy and the culturing, and then what about the biology, the unknown part of the biology of some of these fungi.
While working at Harvard, you have done a lot of work on like fungal species or collections from today, and also the history of fungal collections.
Right.
And what inspired you to focus on that historical side of mycology?
Well, I was appointed curator, so I was responsible for taking care of collections. And those collections, well, the oldest of the fungus collections are about 200 years old. And so I kind of had to understand about who the collectors and the describers were, where they might have been, uh, what what their publications were. So it's an outgrowth of I suppose, the responsibility of taking care of all of those collections. And, part of it is so curious because we have collections, for example, from California from 1840, San Diego, 1840. And if you think about California and San Diego and lichen collections, which is what many of these were, you can be pretty well assured that it's very different today than it was in 1840. And so, who collected them and why were they there and what was the motivation for their activities in a particular area? And then, how do those help us think about the world today?
Mhm.
And I often say that as a curator, I'm kind of living with ghosts. There are all these people who went before, who collected, who named, and who examined these specimens. And it's kind of fairness to the ghosts to know who they are.
Mhm.
So that's what really is the motivation. Plus, just being interested in history and how people got to the places that they got when they were collecting, and these sea voyages of the 19th century where it may have been 6 weeks on a ship to get to a destination, and the perils of a new and different place.
Mhm.
So many reasons, but all curious. Again, curiosity.
Do you think that modern technology like DNA sequencing or other advancements had an impact on the way we study or classify fungi from these historical collections?
Well, sure. What sequencing has done is to allow us to look at relationships in a way that we could, in some ways, only assume in the past. I know everybody talks about sequencing now. I certainly made the transition from purely morphology to looking at sequence-based studies. I don't think there's anything that really substitutes for going out, looking at the fungus, studying it, understanding its morphology and its anatomy, and then maybe you can find out something by sequencing and enhance the uh, knowledge that you're bringing to the relationships. But uh, I'm a firm believer in hands-on with the fungus first,
Yeah.
and then thinking about what sequencing can tell you. And again, I mentioned at the beginning, when I started working on the P's Isales, we didn't really know much about the biology at all. We know now through sequence studies that many of these are mycorrhizal. They're connected with the roots of plants and they aid in the uptake of nutrients and so forth. We only really learned the extent of that once sequencing came into it became a possibility. So it's really wonderful to be able to have that kind of information. But real hands-on study and knowledge is important.
So you have worked at Harvard for many years and witnessed the field evolve, just like you said. And what was it like to watch mycology change over time? What are some of the most exciting or surprising developments you have seen?
Well, I mentioned this idea of mycorrhizal studies, and we knew a lot of that with the basidiomycetes, the Basidiomycota. We knew that they were mycorrhizal. We knew a lot about their relationships. In terms of the ascomycetes, we knew relatively little, and within the Pizizales, relatively little. So that certainly has been a certain kind of direction. I think the other thing to think about these days is the endophyte studies, the study of fungi that are living in living leaves without causing symptoms of disease within those leaves or within those plant tissues. And again, we didn't know much about these, but we do now. We know that because of culture studies and sequence studies, and we know that a lot of fungi that we might not have uh, suspected would be endophytes are in fact living in living plants. And one example of that, again, it's from uh, things that I'm interested in with the Peace Isales. Ascobolus is a genus with species that live on the dung of animals, herbivores primarily. And we know them from picking up the fungi out in the field. But what we didn't know is that some of those are living in as endophytes. And why would we suspect that? We wouldn't. But we're able to discover that with environmental sequence sampling and so forth. So I think that it's really in the biology of fungi that the sequence work has made really great advances and tells us an awful lot about how it is that those fungi get along in the world. Of course, fungi are everywhere. Um, and we know that we can isolate them and pick them up from all sorts of substrates, but that doesn't tell us what they're doing often.
My next question is specifically for my audience. So many of my readers are enthusiasts or early learners, but not professional scientists. So how do you see the role of amateurs in the future of fungal research? Like, what impact can they have? in the field.
Well, I think again, I'm one person. If I go into the field, I have a certain eye for certain fungi. I'll pick up certain fungi and so forth, but I am not necessarily looking for all fungi. And one of the things that forays and amateurs do is that they can go out and there are many of them, many sets of eyes, and they can see things that an individual person might not see. So I think part of it is here it is, we can do surveys and overviews of areas and learn about all of the fungi of an area in a way that an individual can't do, and that as a collective that can happen. We have all these tools now, Mushroom Observer and Encyclopedia of Life and all of those. And those, which are primarily the ideas, they're primarily being populated by amateurs, by non-professional folks. And that, again, I often, if I'm studying a particular fungus, I'll look and see who's found this, where's it been found? And those kinds of records can often be quite important for us to know about. And then I know that sequencing has become much easier, that on many forays and many individuals are sequencing kind of on their own and posting those. And again, that augments what we know and what we can find out about distributions and taxonomy and placement, all sorts of different things. So I think there's a great role these days for individuals who are just curious.
Mhm. And what advice would you give to young people like me who want to learn more about mycology or maybe even do research in the future?
Yeah. Well, see if you can find a good mycology course
Mhm.
and start that way. Then I think you probably these days need to think carefully about how you want to undertake a study. Studying the taxonomy of a group is probably not necessarily a great employment future, but studying the biology of fungi and some of their applications probably is. And so, again, I think I keep coming back to curiosity. Part of what you're wanting to do is driven by curiosity, and you should let your curiosity carry you in in the direction you want to go. I began this all kind of by accident. Uh,
Mhm.
I didn't have a master plan. I thought, I'll study plants. Maybe I'll teach biology in high school. Did I discovered fungi. If I wanted to study more about fungi, I needed to go to graduate school. And then the choice of what I was going to study became kind of accidental too. It was something that I'd collected. I'd looked at these, thought this is a good project, so I'll do it. So curiosity.
Thank you very much for taking the time to speak with me today. I really enjoyed learning about your experience and your insights, and I believe that all this information will be very valuable to my audience as well. Thank you so
Great,
much.
great. Thank you. It was a pleasure.